Okay.
Good afternoon, everyone. Welcome, welcome. All right. My name is Jen Myers, and I'll be the moderator of this session. Welcome to Modern Approach to Microbiology, presented by Lance Shaner. Lance, how are you doing?
I'm good. I'm glad to be here today.
Awesome. Well, thank you. Before we begin and learn some awesome stuff here, I just want to say thank you to Friday's presenting sponsors, Yakima Chief Hops. And I also like to thank Friday's session sponsors, Fraunhofer Design, Surpass Chemical Company, and Deschutes Beverage Technology. Thank you so much to our allied members for supporting us. We wouldn't be able to bring you this virtual conference if it wasn't for them. Also, if you guys have any questions throughout this presentation, we'll be taking them. Please put your questions in chat or use the raise your hand function. And other than that, let's learn some stuff about microbiology, Lance.
All right, thank you, Jen.
Yep.
Glad to be here today to tell you a little bit about kind of our approach to microbiology, and I think approaches you can that are accessible to bring into your brewery. Just real quick, some credentials: microbiology bachelor's from University of Illinois, and a PhD in microbiology from University of Texas in Houston, where that's where I kind of started to study yeast, not in the brewing context, though I was, an avid home brewer at that time, but it was more in the human health context. Took a sharp turn from there and went to law school, but it was actually related. So I wanted to be a biotech patent attorney. So after law school, I became a patent attorney at Marshall, Garcia & Boren in Chicago. Did that for about 4 years until I got tired of that and got the lab bug again and just wanted to be, around beer. So founded Omega Yeast in 2013. So that's my meandering path to yeast lab ownership. Okay, so an outline of what I'm talking about today. Start out by talking about media. So if you've had any, background in microbiology or in brewing microbiology, some of this is going to be, you've heard it before, but if you haven't, this will be new, and hopefully I can get it across in an understandable manner. But the first part, I'll be talking about microbiology media and what it means by selective and nutrient and differential media, and the organisms they detect. Talk a little bit about where in the brewing process you'd use these types of media. Then give you a handful of insider tips for cost-effective equipment and QC methods. These were kind of things we've cobbled together over the year from starting a yeast lab and trying to do it, with my own money in a cost-effective manner. Some of the tricks we've learned to kind of keep costs down for some of these QC methods. Because one thing you may have noticed, if you're ever buying anything from a science supplier, it seems outlandishly expensive, and it is. So if you can find essentially the equivalent items that are being sold to people in a non-scientific context, you can find these items much, much cheaper. So I'll give you a handful of examples of that. And then finally, I'm going to tell you about some of our R&D that we did here at Omega Yeast, looking into diastatic S. cerevisiae. And actually, some of this, I should mention, some of this research happened in New York. That was done in cooperation with a colleague of mine at Cornell University, Pat Gibney. We went to graduate school together and then collaborated on this research into diastatic S. cerevisiae. Okay, so the basics of QC microbiology are essentially these three types of media. So nutrient media allows the growth of essentially anything. So it doesn't necessarily tell you what is what, it's just anything grows in that. And so malt, agar, or YPD would be examples of that. A selective media contains compounds or a compound or compounds that prevent the growth of most of the organisms, but allow for the growth of whatever you're targeting. And an example of this is WLD, which has cyclohexamide in it, and cyclohexamide kills or inhibits the growth of brewing yeast. So if you have a pool of millions of brewer's yeast and say 10 bacterial cells, you plate that on nutrient media, you are not going to see those bacterial cells because it's overloaded with yeast. But if you put that on something like WLD that has cyclohexamide in it, prevents the growth of the yeast and allows for the growth of the bacteria. So you can visualize the bacteria, even though there's 10 of them in a field of millions of yeast cells. Similar with, it actually allows Brettanomyces growth too. Brettanomyces yeast are resistant to cyclohexamide, so that's a way for you to select for growth of Brettanomyces. Another example is LCSM, and this is what used, and I'll tell you a lot more about this towards the end, but you can use this to select for growth of diastatic S. cerevisiae. And strangely, the way this media works is it's got copper sulfate in it, and that inhibits the growth of most ale and lager yeast, but for some reason allows the growth of diastatic variants of S. cerevisiae. I say for some reason because copper has nothing to do with being diastatic. It's really just kind of the strange correlation that diastatic S. cerevisiae happen to be resistant to copper. But that trait allows allows us to use something like LCSM to select for the growth of diastatic S. cerevisiae. Finally, then there's differential media. And this usually contains something that allows one to distinguish between microorganisms by visual observation. And I've got a couple of really good pictures of what I mean by that. And a good example of that media is WLN, which is similar to WLD. They're really the same thing, except WLD has cyclohexamide in it. So WLN is WLD without cyclohexamide. But it's got a dye in there called bromocresol green. And this dye is metabolized differently by different yeast strains. Again, not known why it does this, but it allows you to really see the difference between different strains of S. cerevisiae. And so it's a tool that you can use to see if you have a mixed culture. So, say you contaminated your lager yeast with your with a Belgian ale yeast. It's probably going to help you see that. So this is kind of like this is a little picture I drew up to kind of show you what I mean by all of these concepts. So what you're going to want to consider here is the key is over on the right. So we've got an ale yeast shown in a pinkish orangish color, green diastatic S. cerevisiae, yellow is Brettanomyces, and purple, I think, or black is Lactobacillus. And the dots basically are just if there's a colony, so a cell was deposited on that spot and grew up. So in something like malt agar, which is a nutrient medium and allows for the growth of anything, you get a spot, or wherever that cell landed, it grows. Everything's growing, nothing's inhibited, because that's a nutrient plate. LCSM, which is selective, what you'd find is that only the diastatic S. cerevisiae should grow. So you see, where you had the dots on the malt agar, you see if you were to replica plate this on top of LCSM, only the dots where the diastatic S. cerevisiae was deposited would grow into colonies. So that's a selective medium, selects for the growth of diastatic S. cerevisiae. Some media like WLD are actually a combination of selective and differential. And so if you plate, again, replica plate this onto the WLD, you'll see lactobacillus growth and a ring of clearing or yellowing around it. And that's because this bromocresol green dye is also responsive to pH. So when the pH drops, it turns from bluish green to green to yellow. So you'll get a yellow ring around this bacterial colony will tell you that you have an acid-producing bacteria. Brettanomyces will also grow on this type of media because it really also kind of functions as a nutrient medium. And it grows because it's resistant to cyclohexamide. So in this case, diastatic cerevisiae is not going to grow because it is not resistant to cyclohexamide. Ale yeast is not going to grow because it's not resistant to cyclohexamide, but both Brettanomyces and Lactobacillus are. So that's the selective aspect of it. And the differential aspect of it is that colonies of acid-producing bacteria get this ring of clearing around them from the drop in pH. So this is the concept behind these various types of media. So in the real world, this is an example of the utility of WLN. So we had a customer that does all of their own propagation in-house, but they had reached out to us because they were getting some strange results where they're starting to detect just by sensory some phenolics in their beer that was not supposed to be phenolic. They weren't using a Belgian yeast, just using a clean American ale strain. So I said, go ahead and send us a sample, because the first thing I want to do is try to plate it on WLN. A lot of brewers seem to think that you can mutate like a clean non-phenolic ale strain to a phenolic ale strain. And while it is technically possible, it's exceedingly rare. So, I mean, so rare, I shouldn't even say it's possible. You're not going to see it. Because even if it happens, you're talking about one cell in the midst of, millions or billions of other non-phenolic yeast, so it's not going to be noticed. So more than likely, you've got a mixed culture if this sort of thing happens. So, and again, like I mentioned, WLN is a really, really great medium to do this. So you take a sample of the yeast out of that fermenter, and you streak it onto the plate and let
Oh no, I think Lance froze up.
cells that we saw in their stock culture of their house strain. You can see all these white colonies popping up. So it was pretty apparent right from the beginning that they had a mixed culture here. Interestingly, some of the other things that came out of this were very strange, because they have in-house an invisible sentinel, which can be useful for detecting diastatic yeast genetically. And it was saying it was STA1 positive, but it wouldn't grow on LCSM, which generally allows for the growth of diastatic cerevisiae, but it did grow on FPDM, which is another medium used to detect diastatic cerevisiae. So this was kind of a strange set of results, and they had the same thing in another fermenter, and you can see that on the right. So it didn't have nearly as much of the offending strain in it, but still they were detecting phenolics and banana flavors they don't normally get from their house strain. But this time it tested negative for STA1. That might have just, again, some of these things are still mysterious, but the point of this was really that WLN is a very useful medium for looking to see if you have a mixed culture, just because yeast different strains happen to metabolize this dye differently. So you can see, their house strain pulled in this dye, so the colonies were green, whereas the white colonies either aren't taking up the dye or are taking up the dye and processing it into something that's not colored. So it just allows you to tell the difference between different strains in a mixed culture. Oh, sorry, I was hoping that wouldn't happen. This is another example. So I've heard that some of you guys have heard a talk earlier about kvike. And kvike, as you know now, are farmhouse yeast cultures from Norway, and tend to be mixed cultures. So they have lots of different strains that are all genetically related on some level. And this is what those look like when you streak them out on WLN. You again see this effect where different yeasts take up the dyes differently. So you get some that take up the dye and turn green, some that are white, some that are pale green with a white ring. You just get a wide variety of phenotypes when you streak these strains out on WLN. So it makes it an incredibly useful tool to determine if you have mixed culture. So WLN is great to have around in the brewery if to test your pitching yeast, just to show yourself from generation to generation that everything still looks the same. I haven't picked up anything else that might be pushing flavors in a direction I don't want to go. It's a stable medium, doesn't need to be used super quickly. So it's just a good and easy tool to have around. So there are, again, if you look in some of these catalogs, there are tons of different media out there. There's kind of a core that we recommend that you have, and that is LMDA, which is a lot like WLN, WLD, but is a little better at allowing growth of some bacteria. So, and again, it's differential, so you can see whether they're acid producing or not, whether they clear the area around them, drop the pH, allows the growth of Brettanomyces. So it's really a good a good media to detect a lot of different organisms. You can use it both aerobically or anaerobically to favor the growth of certain organisms. As I've already mentioned, talked a lot about WLN. Very good for just checking your cultures to see if you have mixed culture or if you have a pure culture. And then finally LCSM, very good for detecting diastatic S. cerevisiae. This one is more, it's a little tricky, and again, I'll go into much more detail about this later. It's a little tricky to use because it's not one that you can make a lot of and use over a month time period, because one strange thing about it is it actually gets more selective the older the plate gets. So it will, I shouldn't say more selective, it gets more inhibitory. So even diastatic yeast won't grow on it if the plates were poured a week ago. Somehow the inhibiting effects of the copper get stronger and stronger as the media ages. So if you want a false sense of security, it can be a very good thing to use. Once it gets a week old, you're never gonna see anything grow on it. So everything's roses. But seriously, it's something you want to use within 3 days of making the plates. So it's something you have to be comfortable with making these plates yourself. It's not, you can't buy a sleeve of plates from a manufacturer and hope it's going to work. I hope every once in a while I'm getting a box popping up saying my internet connection is unstable, which is not normally the case here. So hopefully everybody's still hearing me. Okay.
We are. There's a small part where you froze up, but and then you came right back. So far so good.
Well, that's good. And I said this is not even, we normally have very good connection here, so it's annoying that I'm even seeing that box, but I'll soldier on. There are other types of media that you can use that maybe get you a little more specific coverage. MRS plus cyclohexamide, very good for lactic acid producing bacteria. And that NBB can be used for just hygiene monitoring in the brewery. And finally, this other one called SMMP. This really kind of depends on, this is probably more useful for very large breweries with exquisite control over their oxygen, because this allows for the growth of obligate anaerobes like Megasphaera and Pectinatus. I have, in the 8 years of doing this, only ever heard of Megasphaera and Pectinatus problems with very, very large breweries that have very, very sophisticated oxygen control, because these organisms are exquisitely sensitive to any oxygen. So you'll see these things pop up in like CO2 lines that run around, very, very large breweries, because those are always anaerobic and allows for these organisms to survive. Where there's any amount of oxygen around, they die. So not necessarily one that you're gonna bring into the brewery unless you're, very large and have very, very good oxygen control. That is to say, you're probably not ever gonna see these things become a problem, just because the environment's not gonna allow for it. Okay, so where should I look? There are, depending on the extent of testing you wanna do, you can look everywhere, and frankly that's a very good approach to take if you're well set up for microbiology. So even yeast coming straight from the manufacturer, we welcome people to test stuff coming straight from us, and it's a useful thing to do, testing all along the route here, because if you do end up running into a problem, if you're doing this testing all the time and doing it at all these points, when a problem happens, you're already going to be armed with some information as to where that could have come from. So if you tested incoming yeast and you didn't get any hits for diastatic yeast, or bacteria, anything like that. But by the time you got into the package, you were having, overcarbing, and come to find out there's diastatic cerevisiae. Well, where did it come from? If you're taking samples all the way along the route, you're going to be able to determine if you have a problem on your canning line. Some of the lines might be contaminated, or whether you had a problem in your bright tank, or whether you had a problem in your fermentation vessel. But if you have samples all the way along the way, you're going to be able to rule out certain things and really focus your efforts on cleaning what needs to be cleaned. So for that reason, I mean, I don't think you can sample too much. It's just a matter of how much time and resources you want to put into this sort of thing. And you can do LCSM and WLN, I think, are the critical ones for the incoming samples. During fermentation or repitching, again, WLN just to confirm that you still do have a pure culture. LCSM to make sure you're not introducing diastatic yeast, and LMDA to just make sure there's no bacteria. And the similar setup for the bright tank and packaging. And of course, you also might want to consider, testing from each canning head. Again, if you're finding kind of sporadic contamination, it could be down to a single head. So again, really cannot do too much sampling. It's just a matter of the resources you want to put into it, the hours, whether you're going to have a person dedicated to doing this sort of sampling. So what do you do if you see growth on your media? Well, it depends on what media you're seeing growth on, as far as how much to worry. But you might want to then go from there to do more determinations to figure out what it is you actually have, to see if there's something you need to worry about. So you can do classic morphological characterization, for example, Gram staining or catalase staining, or just plain morphology. What do they look like under the microscope? The rods are indicative of lactobacillus or enterobacter. Whereas cocci are more, little round cells are more indicative of pediococcus. Of course, you should know what yeast look like. They're much larger, round budding yeast. And then you can also do, if you're equipped for it, endpoint PCR or something like Invisible Sentinel is something that you can use to determine, say, you have a yeast growing on LCSM. Is it a false positive or is it diastaticus? And you can find this out by using Invisible Sentinel or just PCR if you're set up for it. Similarly, qPCR, something like the Paul gene disk, for example, can also detect the presence genetically of all of these organisms and tell you which one it is. Finally, you could also you could take your plates that have colonies on them and send them off to companies to actually sample off that plate and tell you what that organism is. So you have a lot of options at this point to figure out what it is, and then take measures from there based on that. So I'm not necessarily going to talk through every box on here, but I think this can be a resource for you or a guide to the sort of things that you could do for a quality program, depending on how big you are and how many resources you want to put into it. So I would use this more of a reference guide to come back to, and again, just our opinion on the types of things that you could do depending on your activities, whether you're packaging in cans or whether you're just kegs, and your volume all plays into the types of tests you do and the scope of your quality program. Okay, so I'm gonna again share some kind of the tips that we've accumulated over the years for just doing a quality program cheaper than, you don't have to buy it from science suppliers, and you can do a lot of things from probably cheaper than you might imagine. As I'd kind of alluded to before, if an item or reagent has a use outside of a lab environment, there is likely a more cost-effective option out there. Lab suppliers like to charge a lot for their stuff, and there are alternatives out there. One really good example, or if you've ever used hot hands in your gloves, that is a chemical reaction that happens with oxygen. So what that means is if you have an anaerobic container like this, and this is actually purchased from a science supplier, but this is another area where you could save money by using a mason jar, for example, as your little anaerobic incubation chamber. But in any event, you can use hot heads. So you just, hot hands, open the container, drop that into the container along with your plated micro sample. And that hot hands will soak up all the oxygen and turn that into an anaerobic chamber. Or you can buy anaerobe packs from a science supplier for $6.40 a pack versus 62 cents a pack if you get hot hands from Amazon, for example. They are quite literally doing the same thing. But because hot hands are used to put in your gloves, you're not going to pay $6.40 for warming your hands. But a science supplier thinks they can sell these same things for tenfold price increase. So very useful. Frankly, there's no reason to buy anaerobe packs with hot hands out there. Agar is one you may not think of. So Agar is the stuff that it's derived from seaweed, and it's basically what's used to keep your plates solid. So when you boil it, it turns to liquid. When you pour that into a plate and let it cool, it turns into a solid surface on which you can streak your yeast or plate your beer samples. Some strange people eat this stuff. It's actually a food product. So that's why you can, again, find it on Amazon for down to 7 cents a gram, potentially even cheaper if you get greater volumes of it. But again, from a lab supplier, it's close to 10 times more than that. It does the same thing. We've been using the Amazon stuff for years, and it does the same thing. But this is a food grade agar, and we use it in our plates and save lots of money. And then finally, pressure cooker. So in the lab environment, pressure cooker is called an autoclave. And we have plenty of autoclaves. We use lots of autoclaves because we need higher volumes. An example of a type that we use is on the right, Hariyama HVA 110. 110 stands for 110 liters. That's the capacity of that autoclave. But it's an expensive device, $15,000 for that. If you do a lot of autoclave analysis or searching, they can get much more expensive than that. They're very, very expensive things to have around. But if you're doing small scale, sterilization of media, you can do this in a pressure cooker. Effectively works the same way as an autoclave. It gets hot under pressure. So you can get temperatures much higher than boiling water. It'll get up to 250 degrees Fahrenheit. And even spores of the nastiest bacteria will die at 250 degrees Fahrenheit. So it's what you use to really completely sterilize your media and know that you're starting with a sterile source. And you can accomplish the same thing in a pressure cooker. I really like, and again, you can get very cheap pressure cookers, especially probably if you check Craigslist, something like that, or eBay. I really like these more expensive models, these all-American pressure cookers, because they don't have a gasket. So they basically have, they twist on, it's kind of a metal on metal contact. So basically these things never break down. They're extremely reliable because, gaskets can fail on the types that have gaskets. But this thing is a rock, and it will work for you for years. So before I ever had an autoclave, I had an all-American pressure cooker, and they work very, very well for a lab on a budget, or doing stuff at home, doing microbiology at home. They're very useful. Incubators. So again, extremely expensive if you get them from a science supplier. We've got some pretty handy people around here. So one of our guys built this incubator, which is built from plywood effectively and lined with reflective tape inside, and then we have basically like a terrarium type light bulb to provide heat, and that's connected to a external thermostat. So if you want an incubator to hold at 90 degrees Fahrenheit to grow your bacteria samples, this works really, really well. And it's it was like $200 worth of parts to put this thing together. Or you can do the same thing even cheaper with just a large Tupperware container from Target, something like that, and do the same thing where you put this, you line it with some reflective tape and put a light bulb inside it connected to a thermostat and hold the temperature wherever you want it. So you can make very, very cheap incubators that are very, very effective for the purpose. Now this is not, Whirl packs are not an example of something that you that have another purpose outside, but they're very, very cheap and a good addition to your quality program. And I mean, they're cheap enough that they're really great to use for basically taking samples all along your wort production line. So right out of your wort chiller, once you put it in the fermenter but haven't pitched your yeast, you can pull a sample of this and you swing it closed, and if you've done everything right, you see no growth whatsoever. So it just looks like sterile wort sitting there. Even if you do see growth in this, it's not the end of the world, because a lot of times it's going to be a non-beer spoiler. So it's going to be something that's present in extremely small amounts and that's easily outcompeted by yeast, and will actually not necessarily even show up on later plated samples because it just gets completely outcompeted by yeast. But it's a very, very good thing to use as kind of an indicator of your sanitation. If you regularly use these and you never see growth, you're doing a really good job getting everything sanitary all the way along your wort transfer lines. And again, very, very cheap. So there's not a great reason not to do something like this. Okay. Oh my gosh, I skipped all the way to the end. Okay, I don't know how I did that. So another one, which you've probably heard of, forced fermentation. Really, this is used for testing the fermentability of your wort, and all you need for that is a flask, a stir bar, and a stir plate, shown here. One thing I'm not sure people know is that you shouldn't necessarily always expect your final gravity to match what you see in your forced fermentation. I think the forced fermentation is really ideal to just make sure your wort is fermentable. So it can, if your forced fermentation, if you had 10 Plato starting wort and your forced fermentation stopped at 5, then that'll give you an indication that your wort is not fermentable. But if it, finishes at 2.5, don't necessarily assume then that your batch is going to finish at 2.5, or don't be alarmed if it doesn't. An example of this is we have in our collection some diastatic strains that, if you use them in fermentation, are just ridiculously attenuative. They'll finish over 90% apparent attenuation. But when we grow them aerobically on a stir plate like this, they don't. They'll actually, just a kind of a typical non-diastatic ale yeast will actually finish with a higher attenuation than this strain does. We don't know exactly why. I mean, our assumption is that there's something about growing aerobically stirred like this that doesn't activate its diastatic capability, so it just doesn't attenuate as much. So again, don't be alarmed if your actual fermentation finishes considerably lower or slightly higher. I don't think forced fermentations are great at telling you exactly where something should finish, but they are good and helpful at testing, fermentability and just determining if you have a, major fermentability problem. You actually were mashing at 170 and you thought you were mashing at 156, something like that. So, and obviously, so as far as like setting up a program for, sensory training, there are kits out there, like Flavor Active on the right, that give you samples of a lot of different compounds for training yourself on detecting these in beer. And those can be a little costly. Or, you can use lower expense techniques to kind of induce the same thing, like vinegar, instead of buying, getting acetic acid in a kit, or letting your beer sit out in the sun for a little bit to get that sunstruck aroma, or imitation butter to give you the diacetyl. But there are, again, even in this context, cheaper methods for doing sensory analysis, or training yourself on sensory analysis. So lots of places you can get this stuff. Consistently, the more expensive outlets tend to be places like Fisher or VWR. They do have, there are certain things that we get from those outfits that you just, that are very specific to labs. So you can't necessarily escape the science suppliers, but and Weber Scientific has always been very good too. They have pretty reasonable costs for a supplier. But always check Amazon too, or just search the internet, or go to the grocery store. There are certain things you can get at the grocery store that are useful in the lab. Okay, shifting gears again, I'm going to talk about our R&D in the diastaticus realm. And again, this work was conducted in collaboration with the Gibney lab at Cornell. So I've been talking a lot about diastatic yeast, and most of you probably know generally what they are. But a quick review of that is they are, genetically basically Cerevisiae. They're ale yeast, but they have a capability that other yeasts don't have. And that is they secrete a glucoamylase, and that's encoded by the STA or STA1 gene. And what this amylase does is it breaks down dextrins and starches. So it cuts off glucose units from a longer chain dextrin or starch. And when it does so, it makes that glucose unit available for the yeast to bring into the cell, ferment. And when that happens, and you don't want it to happen, you can get exploding bottles. So again, these are widely used brewing strains. Like these strains are strains we purposefully use. Certain Belgian yeast, Saison yeast, some Weizen yeasts are diastatic. This is, it's been an issue in the brewing industry long enough that all the providers that I'm aware of, and we certainly do, make it known when you're looking on a product, whether or not it's a diastatic yeast. So it is something you're going to want to know because you're taking some amount of risk by using these strains in the brewery. Because if you were to cross-contaminate your cultures, and you don't notice it during fermentation, you go to package, you could have problems with overcarbonation. Now, overcarbonation is not always due to diastaticus. There are other things that cause that, hop creep, outside the scope of what we're talking about here today. But one of the things that can cause the problem, cause a big problem, is diastatic Cerevisiae. Okay, so there's enough about the background there. So I'd already mentioned that LCSM is useful for detecting diastaticus, but one of the things we found is when we were buying our LCSM from Weber Scientific, it was extremely stringent. Like even when we test known diastatic yeasts, we'd not get growth. So as we started to do a lot of research on this, and we have three groups of yeasts here, and one of them, the group one here, is STA1+, so it's diastatic, and it has this, again, I'll talk a little bit more about this, but a promoter element that allows for that gene to be expressed at a high level. So all these strain numbers shown here, they're all growing on again, this is a nutrient medium, so a medium that allows anything to grow. We have a second group here that is STA1 positive, but has a kind of a broken version of the promoter that makes it not quite as diastatic. Again, go into more details in future slides. And then group 3 are strains that we found regularly showed up on LCSM, but are not diastatic. So we call those high background strains. So those are just false positives when we get them on LCSM. And this goes back to what I told you at the very beginning, is that this growth on LCSM is based on copper resistance, and copper resistance isn't necessarily, equated to diastatic activity. It's just this strange correlation that most diastatic yeast happen to also be copper resistant. That doesn't mean that you can't be copper resistant and not diastatic. There are these high background strains are those type of strains. They're not diastatic, but somewhat copper resistant. So some limitations of this type of media: false positives. So again, some non-diastatic strains can grow on it. False negatives. Different strains are sensitive to different amounts of copper sulfate. And again, the degree of growth is not correlated to the degree of attenuation. It's just correlated to the amount of copper resistance. And so this can be a reliable method, but it requires a lot of optimization and validation in your brewery. You're going to want to have some kind of positive controls to make sure your medium is behaving the way you expect it to. So one of the interesting things we saw is that, again, this illustrates the effect I was telling you about before with age on plates. So the older the plates get, the more restrictive to growth they are. So these group 3 high background strains, by the time you get to plates that were poured 96 hours before we put yeast on them, you see less of this background, but you also see less growth of these actual diastatic yeast than you do on 24-hour plates. Similarly, this other medium sold by Weber Scientific that's used for diastaticus detection, it's even more prominent with that. So you can see if you're looking at 96-hour plates, nothing is growing. Even the strongly diastatic yeast, nothing is growing by the time you get to 4 days out. So again, you have to be mindful of this when you're using these kinds of plates. If you're using a week-old plate and you see no growth, that does not mean that you don't have diastaticus. So it can be important when you're using these plates to kind of always have a control around something that should grow on these plates. This really kind of because we saw this effect where known diastatic strains were not growing on LCSM that we purchased from Weber Scientific, is what drove us to kind of formulate our own LCSM. I mean, it's a published formula, but you can tweak some of these things. Like you can, increase the amount of copper sulfate in them or decrease it as necessary and kind of tune it into detecting what you want while inhibiting the growth of non-diastatic yeast. And the reason we did this is because we saw this regularly with purchased LCSM. It really didn't give us a huge amount of confidence that we were always going to detect if it was there, because even in fresh plates, there are some diastatic yeast that wouldn't grow, that we knew to be diastatic. So again, keep that in mind. That we've published this work, and you can read a lot more details about it in that publication if you'd like, in the Journal of the American Brewing Chemists, Journal ABC. Okay. The current genetic methods that are used to detect diastaticus have some limitations. You can get false positives. We have a really good story around false positives. We had a customer that would buy smaller pitches and then prop those up into, by the time to get to 100-barrel pitchable. And this is a brewery sophisticated enough to test everything that's incoming. So they would test the cultures we provided them. They test clean genetically, no diastaticus. They do a first stage prop, no diastaticus. They do their final step up and then make their beer. And in their beer, they would buy gene disc from Paul. They started to detect diastaticus. And this happened through multiple independent pitches. And so they came to us with questions like, how could this be happening? We are detecting, or we aren't detecting it in what we're getting from you, but by the time we get to this last stage, we are. And we're really kind of banging our heads against the wall with what could have been happening. And then they remembered that they had seen really this exact same fact pattern multiple years before with a completely different yeast and a completely different supplier. And somebody finally put 2 and 2 together that they were using the same additive for those 2 beers. The one they did multiple years ago was a wit, where they wanted some haziness, and then this current beer was in a hazy IPA style, and they were using an additive to kind of help give them the stable haze. It turns out this additive is a yeast-derived product, and what they found when they tested that yeast-derived product itself is that it would light up their genetic tests for Diastaticus. It wasn't really an issue, or they were never seeing overcarbonation here, because this is something that was added to the kettle, so it was boiled. But the point is, these genetic tests test for DNA, and you can have boiled dead yeast in your beer, and if it's has the STA1 gene in it, these genetic tests are going to detect that. Even though there's no live yeast, there's nothing to worry about. So this really kind of shows you the limitations of some of these genetic methods. They're not testing for live yeast. They're testing for the presence of the STA1 gene. You can have the STA1 gene there without a live yeast cell. So this is really, I love this story because I think it gives you a good indication that you need to, that all these methods work kind of in tandem. I don't think microbiology is the be-all end-all because it takes a little longer. You have to wait for things to grow on these plates, whereas the genetic tests are very, very fast. You can have a result in a few hours. But again, the genetic results have flaws too, in that there might not be live yeast there. So these methods really kind of complement each other. And if it's up to me, you use both of these methods because they both have utility. As I mentioned before, there was a publication a couple of years back that showed that even in diastatic yeast, so yeast that were positive for the STA1 gene, it's been known for a long time that these different yeasts have different levels of superattenuation. And this paper kind of took a step in the direction of figuring out why that was. What they found was that there was a sequence in the promoter region. So for those who aren't geneticists, this is a part of a gene that basically tells the gene when to be turned on and how much to be turned on to actually make that enzyme. So what they found is that there were some diastatic yeasts that had a portion of their promoter that was deleted. And the deletion of that promoter element correlated with the amount of diastatic activity. So if they had a deletion here, they were less diastatic. They'd chew up less starch. So that was helpful. And this is kind of a depiction of what you see here. So strongly diastatic yeasts have this upstream activation sequence, which is what UAS stands for. And so they make a ton of this enzyme, and they can break down all the starches present. So these are highly diastatic yeasts. Weakly diastatic yeasts have this broken version of this upstream activation sequence. They make less of the enzyme, and they're not quite as super attenuative as the other yeast. And of course, a non-diastatic yeast does not have STA1. Doesn't matter what their promoter is because they don't have STA1. You don't get breakdown of starches and dextrins. But it was clear to us from a lot of anecdotes internally that this wasn't the entire story. So we find a lot of cases that the amount of attenuation does correlate to whether it has a deleted promoter or not. If the promoter is intact, they tend to be more diastatic. But even within these breakdowns, there's a gradient of the amount of diastatic activity. So it's not telling the whole story. So in our mind, it's still useful to have tools that can kind of differentiate more between these variable levels of diastatic activity. So in this experiment, we took all of our known diastatic yeasts and got some Lagunitas IPA off the shelf, snapped open the bottle and squirted some diastatic yeast in it and stored it at 75 degrees Fahrenheit for a month, and then measured the Plato and the alcohol. And what we found is there's a pretty good range of, again, these are all, except for these first two samples, all of these are STA1 positive yeast. and you see a pretty wide variety in the amount of super attenuation you have here. And consistently, this 026 is always the most diastatic. It's French Saison strain. It's extremely diastatic all the time. There was a pretty decent grouping of the amount of attenuation and to the promoter status. So those that had the deleted promoter that aren't as diastatic, they'd cluster around a kind of less diastatic range. But again, you see a spread here. And same thing with the STA1 positive yeast that had an intact promoter. Again, you see a wide variety of super attenuation in this bottle experiment. So this goes to show you that the promoter status does not tell you the entire story as far as how diastatic a yeast is. The amount of yeast that are contaminating can have an effect. So this is basically the same experiment with either 10 cells per mil, 100 cells per mil, up to 10,000 cells per mil of these 4 strains. And you can see that, so like OIL 027, for example, has a broken version of that promoter. And even at the very, very high level of contamination, doesn't have a huge effect on attenuation. It does start to have some, but it's not remotely as high as something like 026 or 056, which both have promoter intact, so they're strongly diastatic. But even with these, you still see a dose effect. So the more yeast you start with, the more attenuation you get. Interestingly, this 042 is a promoter deletion. And it, when you get to a high enough level, even though this has its promoter deleted, you still do see some super attenuation with that strain over time. So our goal with this was to develop an assay that provides a readout of diastatic activity. So it gives you an idea. So some tests that you can do yourself that will tell you whether a diastatic yeast you have is one that you have to worry about, one that's going to has a lot of diastatic activity. And this was what we think is kind of a clever assay. And we took a yeast strain that I'd been using all the way back to my graduate school days. This is just a common strain used in the lab that has it accumulates a pigment, a pink pigment as it grows. And so the idea of this assay is you take a plate that has dextrins in it as the only sugar source for it. So the only thing that's going to be able to grow is a yeast that makes this glucoamylase can break down the dextrins and supply glucose to the cell to grow. And if it does it really, really well, it'll secrete this enzyme into the medium. It'll start to break down all the dextrins in the area, and it'll actually allow it to feed the cell, the cells that you plated next to it. So, and you can see this by a pink patch growing up is an indication that it's being fed by the diastaticus strain next to it. So the more diastatic you are, the more kind of red cell growth you get from the cross-feeding. And that's in fact what you see. So in the control over here is a non-diastatic strain plated next to the indicator strain. And here, there's a wisp of growth that you always see just because there's always some amount of contaminating sugars in agar that allow just kind of a little bit of background growth. But you're not seeing any pink growth. But when you plate this indicator strain next to these strains that have the UAS intact, so they're highly diastatic, you start to see robust pink growth here. And it's because this yeast is pumping out a lot of glucoamylase. It's breaking down starches and providing sugars to the colonies next to it. So you see pink growth here. And then you see a lot more variation on the side of strains that have the promoter broken. So, and you kind of saw that in the assays I showed you before. There's more of a spread of the diastatic activity with these, and you see that here too. So something like 027 seems to have a little more cross-feeding capability compared to something like 040. We actually later found that 040 has what looks like a mutation in the glucoamylase gene itself that inactivates that gene. So it's not diastatic, even though it comes up as STA1+ genetically. Which actually, as an aside, then again shows you another weakness of the genetic tests, because they're not differentiating between, a broken version of the STA1 gene versus one that produces a functional glucoamylase. But this assay, which depends on cross-feeding, will tell you whether you have one that actually works. So this was kind of a surprise that we discovered just from doing this assay, is that there are versions of these STA1+ strains out there that aren't diastatic. So there's a lot more, and if you're super interested in this topic, I'd recommend seeking out that journal article that we published. There are more assays, including some ones that were developed by our colleagues at Cornell. So the conclusions from this work is that not all diastatic strains of S. cerevisiae pose the same risk. And the assays we developed can kind of do a little more to tell you what the risk level is if you happen to find something that has a diastatic contamination in it. And could also go to explain why, maybe sometime in the past you detected it in there, but you never saw overcarbonation. And I mean, this is the reason. It depends on which strain is contaminating, depends on how much of that strain is there. So just the mere presence of something doesn't tell you the whole story. Conventional detection methods like PCR and selection media do not provide a readout of diastatic activity, and we provided some assays that do. So some of these functional assays can help you make a more informed decision on whether to destroy product or issue a recall, and just kind of know what risk you're dealing with. And obviously, if you're in doubt, call your yeast provider, and they can guide you along the way. So with that, acknowledge the folks at Cornell, as well as our crew that did a lot of this research as well. And with that, I'd be happy to take any questions, and contact information if you ever want to reach out.
Awesome. Thank you so much, Lance. That was great. We have just about a couple minutes left. If anybody does have any questions, feel free to unmute yourself and go ahead and ask. All right.
I have a question.
Go ahead, Tony.
Is the only way to really do that micro is to make the media yourself?
The LCSM, absolutely. There's really not an option for you to buy plates and use them, because the window of use, the window of utility is so short.
Right.
But there are, there are places. I mean, we do some of this. It's not a huge part of the business, but like LMDA plates are stable. WLN plates are stable. If you want, you can have people make those, make a sleeve of plates and send those to you. So there are some options for that.
Okay, and then you talked about autoclaves a little bit, and we're kind of moving into a direction where we need to take a little bit more care when it comes to micro. We don't really do any micro right now. We only do cell counts and that kind of thing. But like I boil the stones and I boil the Zwickels. But is there any other places that I should be really careful with as far as like making sure things are super sterile that you can recommend?
I mean, anything that's going to contact the wort,
Sure.
Do the best you can. If you do get set up for like a pressure cooker or anything that'll fit in a pressure cooker and survive a pressure cooker, good to throw those things in there. So it really depends. If it's small enough, try to do something like that. If it's not, then you have to do, the best you can with like a CIP sort of thing, just running, like hosing, running chemicals through it to get that way, and just inspect things, make sure your hoses are in good shape. Just, all the little things.
Very cool. Thanks. Great. Awesome seminar.
Thank you.
Any other questions? All right. Well, thank you so much, Lance. This was great. We really, really appreciate it. If anybody does have any questions that come up for Lance, please contact him. He's a great resource for us. So thank you all again, and we hope to see you at the next session.
Thanks, everybody.